Hello everyone, this video includes some places to find information for any last minute revision you want to do before the exam. First of all, I've included some information on where to find a list of the topics that is on the Biology Paper 1. If you look in your arbor on your assignment, you'll find an assignment that was set from me before half term. On there, it includes the list of topics that's on Biology Paper 1, which you can see here on this slide. It also gives you a column to tick off and you can rate them whether you're confident or not very confident on any of those subtopics. And also it includes a PowerPoint with links to individual revision videos that you can then just click on and it will take you straight to that subtopic. I've also included some common errors to avoid, which is what we've seen when we've marked the mock papers in previous years. So first of all then, please make sure you read the question very carefully and identify the command word. So for example, is it asking you to describe, explain, compare or evaluate? Describe, say what it looks like. For example, it could be the shape, the size, the temperature. If it's talking about the line on a graph, does it increase or decrease? And any changes that you can see if any diagrams are given. If it asks you to explain, say how and why. This happens because. Use comparative language if it's a compare question. Make sure that you think about your technique before you start to answer. For example, are you going to compare point for point, or are you going to do a paragraph talking about one aspect and then a paragraph talking about the other aspect? Make sure you use comparative language. For example, this is this, whereas something whatever is the other, or things like however. Finally, if it asks you to evaluate, make sure you talk about the advantages and disadvantages, and most importantly, at the end, make sure you include what you think and why. For example, what you based your opinion on. And always use the every method when you have any kind of calculations. If you make a calculation error on your calculator, then if you've shown your workings out, you'll still get partial credit for selecting the right equation. And common errors to avoid on graph tasks. Generally, they ask you to do at least 3 things. So make sure you read the question carefully and you tick off the tasks as you do them. They generally ask you to plot the data from a table, label the axis, which you can get those labels from the labels of the top of the table, and then draw a line of best fit. Once you've plotted your points, don't include any anomalies that stand out from your line of best fit. Bypass them. Don't do a little mountain or molehill to incorporate them. It should be a straight line of best fit. Make sure you interpret the scale on both axes before you plot your data. So for example, how many little squares are represented by the scale? And make sure you plot your data and you draw your lines of best fit in pencil. If you make a mistake, you can easily rub it out. It looks better then for the person who's marking it, and they'd probably be more inclined to give you the marks if it's a clear, sharp line. And finally, some other common errors to avoid and things to check before you finish the exam paper. So when you're approaching the questions, check how many marks the question is worth. If it's for 3 marks, make sure you've included 3 specific points. So for example, if it was to describe a graph, what happens at the beginning of the graph, what happens in the middle of the graph, and what happens at the end of the graph. Be specific. Don't refer to things as it or they. That is way too vague. So for example, if you're describing the difference between a eukaryotic cell and prokaryotic cell, as you can see from the example, I've named the one that I'm talking about before I've described its feature. Always check your answers make sense after you've written them. Sometimes in haste you'll write in text speak, but please just go back and check that it is written with good English and grammar. And Finally, I've included some other slides on the video that cover the osmosis required practical, eukaryotic and prokaryotic cells, and respiration, which are topics that regularly crop up on Biology Paper 1. This required practical investigates the effects of osmosis on plant tissue, and by the end of this you should be able to describe how to investigate the effect of concentration of solution on the rate of osmosis, how to calculate percentage mass change, and how to identify the concentration of cell sap inside the plant cells. Firstly, osmosis is the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. A dilute solution is one where you have a high concentration of water molecules compared to a low concentration of dissolved solute. That could be salt or sugar. A concentrated solution is the opposite. This is where you have a low concentration of water molecules compared to the concentration of solute. When cells are placed in a dilute solution, water will move into the plant cells because there's a higher concentration of water in the beaker. In cells that are placed in a concentrated solution, water will move out of the cells because there's a higher concentration of water inside the cells than inside the beaker. In this investigation, the independent variable is concentration. It's always the subject of the investigation. Sometimes they could ask about the effect of temperature on the rate of osmosis, and in that case temperature would be the independent variable, and also they could give you an investigation investigating the effect of surface area, and again that would be the independent variable. The dependent variable is the variable that you measure, and in this case we are going to measure the initial mass and the final mass in order then to work out the percentage change in mass. The control variables are the things that you keep the same to make it reproducible. When they give you a diagram like they have in this question, it's a gift because you can see immediately things that they keep the same. For example, they all have used the same volume of solution. Secondly, you can see they've all used the same size of plant cell. It could be length or it could be mass. You may have to stipulate. Equally, they will have conducted the investigation at the same time, so therefore it would have been the same temperature, and you would have left the cells in the solution for the same length of time. You need to be able to calculate percentage gain or loss of mass in cells. The formula for this is the change in mass divided by the total mass times by 100. The change in mass is calculated by subtracting the initial mass from the final mass. In Beaker 2, we can use the data from the table at the top to work out the change in mass, and then we can use that figure to calculate the percentage change. So change in mass is 12.2 minus 10.6. That gives us a value of 1.6. We would use the change in mass then divided by the total mass at the beginning, which is 10.6, times by 100. You can use your calculator and you would get a value of 15%, and it's a positive number, so therefore it indicates that it gains mass. In Beaker 4, on the other hand, again, we'd use the data at the top of the table, the final mass and the initial mass. So it would be 7.9 minus 10.1. That gives us a value of
minus 2.2. So that's the change in mass. We put that then into our percentage mass formula. So it would be minus 2.2 divided by 10.1 times by 100, and that would give us a value of minus 21.75. The data in the table is only to one decimal place, so we would then round that up to minus 21.8, and that is a loss. If you are asked to plot the data, then it's important that you read the question carefully. It generally asks you to do at least 2 or 3 things. In this question, it asks you to choose a suitable scale and label for the y-axis, plot the results and draw a line of best fit. As you can see, I've gone ahead and written in the scale for the y-axis. Once you've done that, you need to work out then what one little square on the graph paper is equal to. To do this, pick 2 points on your y-axis and count how many little squares are between it. So for example, between 0 and 5 there are 10 little squares. We would then do 5 divided by 10, which equals 0.5. When you're plotting your data, it helps you to be more accurate because you can work out then where numbers such as 12 or 24, which are not on a major grid line, are. Once you've plotted your data, then go ahead and draw your line of best fit using a pencil and draw it freehand. Do not use a ruler. Once you've drawn in your line of best fit, you can then work out what the concentration of the cell sap is inside the cells. This is where your line of best fit crosses the x-axis here. And again, you would have to work out what your scale is going across the x-axis. So again, we would pick 2 points on it. So between 0.2 and 0.4, we have 10 little squares. So this time it would be 0.2 divided by 10, and that comes to 0.02. So we can work out precisely then where our line crosses, and we can see at 0.4 it goes one little square over. So therefore our concentration of cell sap inside our cells is 0.44 grams of sugar. In order to explain mass gain or mass loss, you need to be able to include 2 things in your answer. Firstly, the difference in concentration between the cell and the beaker, and then secondly, whether water moved in or out of the cells. Be very careful when you're constructing your answer because you need to make sure that you talk about one or the other. So for example, you either talk about the concentration of water in the cell compared to the concentration of water in the beaker, or you could talk about the concentration of solute inside the cell compared to the concentration of solute in the beaker. In the model answer that I've written on the PowerPoint, I've talked specifically about the concentration of water, and that's in both where they gained mass and where they lost mass. Before you go into the exam, you need to know the difference between eukaryotic and prokaryotic cells. Eukaryotic cells include all plant and animal cells and are therefore multicellular organisms. Prokaryotic cells are bacteria and are single-celled organisms. So immediately you should be able to know with a question like this that the 2 cells on the right, the liver cell and the mesophyll cell, are plant and animal cells and are therefore eukaryotic, and the bacterial cell is prokaryotic. This question only asks for two similarities between prokaryotic cells and eukaryotic cells. There are three differences, so make sure you can recall those three differences off by heart. Just by looking at the images, you can see that they all have cytoplasm, they all have a cell membrane, and thirdly, they all have ribosomes, which are indicated by these small black dots in all three cells. You also need to know the four differences between a prokaryotic and eukaryotic cell. So you can stop the video and you can recap the differences in your own time. This question only asks for three, but there are four differences. When you're writing your answer, think about your technique before you start writing. Are you going to write about how the prokaryotic cell is different from the eukaryotic, or are you going to describe how the eukaryotic is different from the prokaryotic? So make sure you're clear in your answer, like I have been in the model answer on the PowerPoint. Firstly, prokaryotic cells are much smaller, and you can tell this from the diagram. If you look at the bottom of all the images, you can see that the bacterial cell and the liver cell are in micrometers. But then if you look at the mesophyll, it's in millimeters. Millimeters is a bigger unit than a micro unit. And so they've just really put that in there just to test you. Secondly, prokaryotic cells don't have a nucleus, whereas eukaryotic cells do. You can see the nucleus clearly in the two eukaryotic cells. Thirdly, in prokaryotic cells, the DNA is free in the cytoplasm, as you can see by this long strand here. And secondly, it also has DNA in plasmids, which are little loops. And then finally, a fourth difference is that prokaryotic cells have no mitochondria. And again, you can see this on the diagrams. Both the mesophyll and the liver cell have mitochondria, which is always represented by this oblong shape with a little wavy line in it. There are two types of respiration that you need to be able to describe, explain and compare. Aerobic respiration takes place in living cells and uses oxygen and glucose to produce energy, carbon dioxide and water. This type of respiration produces the most amount of energy, and that's what animals use mostly. Anaerobic respiration in animal cells is generally when the muscles become tired and there's a lack of oxygen. And the word equation for this differs because you've only got glucose before the arrow as the reactant, and then the product is lactic acid. Lactic acid is the chemical that causes cramp in muscles, and this would need to be broken down following exercise when you start to breathe in more oxygen, and that oxygen helps to oxidize the lactic acid. This only produces a small amount of energy, and it's not obviously the preferred method of our cells. Anaerobic respiration in yeast is also known as fermentation, and again this lacks oxygen. It starts off with glucose, and when broken down it produces ethanol and carbon dioxide. Ethanol is used in the brewing industry to make alcohol, and the carbon dioxide that's produced is used to make bread rise. You are quite often asked to compare anaerobic and aerobic respiration. So if you can recall the word equations, you can write them at the top of the page if they aren't given, and this will help you then when you're writing your answer. Compare means you would describe the similarities and you would then describe the differences. Before you write your answer, make sure you think about your technique. Are you going to write it in paragraphs, i.e. the similarities and then a paragraph for the differences, or are you going to write it point for point? I would recommend you write it point for point if you find it hard to write a long extended answer in a paragraph. So like I said, think about first of all your technique, point for point or paragraph, and then secondly, make sure when you are writing out your statements, make sure that you specify which type of respiration you are actually talking about. There are two similarities. Both of them use glucose and both of them take place in cells. There are four differences that you need to be able to recall. Aerobic respiration uses oxygen, and a good connective here would be whereas anaerobic respiration doesn't. Secondly, aerobic respiration is used by eukaryotic cells. However, anaerobic respiration is used by prokaryotics. Thirdly, aerobic respiration releases lots of energy, whereas anaerobic respiration only releases a little bit. And finally, aerobic respiration takes place in the mitochondria, whereas anaerobic respiration takes place in the cytoplasm. This would be the information that I would use on a flashcard if you've got time to make any of them tonight. Finally, I'd just like to say good luck in the biology exam tomorrow morning. Thank you.
Lastly, I have included a question that crops up quite regularly concerning the practical in B3, communicable disease. And this was where students set up an agar plate that was infected with a bacteria, and different antibiotics were placed on the agar plate. It was left for 3 days, and when you looked at it following the 3 days of incubation, you could then measure the zones of inhibition, which is the area surrounding the antibiotic discs, as you can see in the diagram here. For example, with antibiotic A, we can see the ring around it, and that is what we would describe as the zone of inhibition. Now, there are two things they can ask you about this practical. Firstly, they could ask you to calculate the size of the zone of inhibition. And in order to do that, you would need to use pi r squared. Obviously, pi 3.14. In order to get the radius, you would then measure the diameter of the zone of inhibition. So if you look at antibiotic D, if we measured that zone of inhibition, that diameter, and it was 10 millimeters, we would therefore put in 5, we would divide by 2, and then don't forget our equation is squared. So 3.14 times 25, and therefore our zone of inhibition is 78.5 millimeters. Now, secondly, they could ask you to describe the results. And there are a couple of things that you can take from your results on your agar plate. First of all, they could ask you to compare the effectiveness of the antibiotics, and you can assess the effectiveness of the antibiotic by the size of the zone of inhibition. But equally, they could ask you in the sense of the resilience of the bacteria. And in this case, then, you would say that the bacteria was more resistant to antibiotics A, E, and D. Equally, another thing that quite often crops up is where they try to catch you out. And if you have a look at antibiotics B and C, neither of them have a zone of inhibition. So it could be concluded that both antibiotics were effective against the bacterium, but equally, if you look carefully, B is slightly bigger than C. And if it's slightly bigger, then that means it has more antibiotic on it. And therefore, if it's a higher concentration, It could be that the bacterium are more resilient at high concentrations, but equally the antibiotic could work at lower concentrations. So I hope that has helped, and good luck on Friday.